Hi everyone, welcome to my channel Pure Biology, and today I come with another video that is the continuous part of initiation process. That is today I will try to explain about the eukaryotic initiation process. Okay, so in my previous video I already have explained about the eukaryotic initiation process of the translation, and here I will try to explain about the I have already explained the prokaryotic initiation process. Here I try to explain that the eukaryotic initiation process. Okay, so here, that is the, in case of initiation of the eukaryotes, the main process is same, but some things are changing here. Okay, here like that eukaryotic cells require more IF means initiation factor. Second, the initiator is here tRNA mate. Okay, tRNA mate. And the third part is that the EIF2 and EIF3 both bind to the 40S or the small subunit. Okay, 40S or the small subunit, and the EIF3 that maintains that free 40S subunit. EIF3 maintains free 40S subunit. Sorry for the noise, because I am just a side of the road, so please adjust the sound, and I am trying to give with the explanation in the proper voice, so please just adjust. Next one, that EIF3 that maintains the free 40S, EIF2 that is the trimeric GTP binding protein. Why this is the trimeric GTP binding protein? Because in the active form of EIF2 that binds with the tRNA imate and also GTP. Okay, here the EIF2, GTP and tRNA imate that all bind with each other. Okay, and form a complex that is called the ternary complex. Okay, and the next one is that EIF3 and the ternary complex that bind with the 40S and convert this 40S to 43S, and these are all only the small subunit. These are only the small subunits. Sorry. Okay, and next step, it is done in presence of mRNA. Obviously, mRNA present there at that time when the 40S is converted to the 43S. Next one is that EIF4F. Okay, here one thing is that E for the eukaryotic. Okay, IF means initiation factor and E for just belongs to eukaryotic. EIF4F that binds to the mRNA. So when mRNA is present there, now EIF4F that binds with the mRNA, and EIF4G that acts as the scaffold protein which links other components. That means other components also added here to make the complex. Next one, that EIF4E that binds with the 5' cap, and cap binding protein is called the EIF4E. EIF4A acts as helicase that unwinds any secondary structure at the 5' end. If any secondary structure is there, so that becomes unwinded with the help of EIF4A. And EIF4B that join the 43S pre-initiation complex. Okay, 43S pre-initiation complex, and through the protein interaction. Through the protein interaction. Next step, the 3' poly A tail also participated in this initiation process that is already occurring at the 5' end. Okay, or you can say that complex formation that is occurring at the 5' end with this 5' poly A tail that participate. Next step, the poly A tail, this is the tail, binding protein, this is called the PABP, the short form, associated with the 3' poly A tail interact with the EIF4G. Here the another initiation factor involved, that is the EIF4G. Next one, this implies the circular organization of the mRNA. That means as the poly A tail that is also involved in the 3' end, so that's why there is a circular structural form is seen. Okay. In the next step, the 43 is that translocates along the mRNA. Okay, an ATP dependent process until it encounters AUG. Which is the ATP dependent process? This 43 is translocation along the mRNA. This is the scanning process. This is the ATP dependent process, and it is continuing till it can recognize the AUG, that is the start codon. Okay. EIF1 and EIF1A that involved here, and the small subunit stops when it reaches to the initiation site, and there forms the 48S ribosomal subunit. That means the small subunit that reaches to form the 48S subunit, and it stops there where the initiation site, at the initiation site. Okay. Next step, EIF5 or EIF5A and a GTPase activating protein that join the growing assembly. That means it helps to join the growing assembly, this initiation factor or this initiation factor. And GTP hydrolysis occur, that is continuing, and GTP hydrolysis occur is done by the EIF2. Now EIF5B that enables the 60S to join the complex. At that time, after these all the steps, now the large subunits, that is the 60S subunits, join to the complex. Before the EIF2 can participate another initiation, its bound GTP that must be replaced with a GTP, because without replacing of GDP by the GTP, the EIF2 cannot participate in another initiation process of translation. So that's why GDP must be replaced by the GDP, and this is done by the EIF2B factor. Okay, so I am just trying to the eukaryotic initiation process to understand you in very simple way. Okay, just steps I have written here, just clearly describing the process, or you can say that if I try to this process elaborately, the steps are so many, so you cannot understand properly. That's why I am trying here to give the explanation of the eukaryotic initiation process of the translation in a very brief and the shortcut, you can say, and very simple way. Just what things are happening in this process I already explained. Okay, so you just read in that way, in the step wise, so you can easily understand this eukaryotic initiation process of the translation. And another thing is that if this video is liked by you, so please you share this channel, and if you are the new one or the newcomer in my channel, so please subscribe this channel for the growth of this channel, and you can comment in the comment section for what I'm saying, that if you can comment in this comment section, so I can understand that this video, in this video I have explained properly or not. Okay, so please you try to comment in the comment section. So thank you, and please continue with my channel.